The principal way that we convey blood around the body is through the blood vessels. And physiologically speaking, there are a couple of interesting phenomena that relate to blood vessels we're going to learn about. And these relate to how they can change their diameter and how this relates to both blood pressure and also the amount of resistance or friction that blood cells encounter in different sized blood vessels. But before we go into the physiology, we need to just get a broad lay of the land of what the different types of blood vessels are, what they look like anatomically speaking, and what their major roles are. So first of all, you probably know most of this, so this is just a quick review for you. But the general anatomy of blood vessels is that in the body we have two major types of vessels, actually three. We have what are called arteries, and these are vessels that carry blood away from the heart. We have veins. By definition, those are blood vessels that carry blood toward the heart. And then there are these very, very small blood vessels called capillaries that I'm going to talk about more in just a second. The generalized route of the blood, regardless of whether it's going through the systemic circuit or through the pulmonary circuit, is that it generally travels through an artery. Remember, again, that's carrying blood away from the heart. It passes through a capillary bed at some point. And then it makes its return journey to the heart through a series of veins, through venous return. Now, there are exceptions to this predetermined pathway of artery, capillary bed, vein. Some of those exceptions you've already learned about. For example, in the endocrine system, we learned about something called a portal system. By definition, a portal system is any time I go from one capillary bed to a second capillary bed without first going to the heart. So again, we've learned about portal systems with relationship to the connection that exists between the hypothalamus and the anterior pituitary. There are other examples of portal systems in the body. The small intestines are connected directly to the liver through the hepatic portal system. And we're also going to see in a little bit when we look at the urinary system that the kidneys contain a number of different portal systems. Other exceptions to the one capillary bed per trip around the body are these things called anastomoses. So an anastomosis, as you'll remember from our discussion of the heart, is a collateral blood flow or an alternate way or pathway of getting blood delivered to a particular location. So imagine if I am a blood vessel, a blood cell down here at this location. I don't necessarily have to travel through this capillary bed. I could, in fact, travel downward through an anastomosis through a different capillary bed. Sometimes there are anastomoses, such as this one right here, where you can completely circumvent the capillary bed altogether, in which case you went from an artery into a vein without going through a capillary bed first. So this is another example of an anastomosis. So there are, anatomically speaking, three different distinct regions of any blood vessel. It doesn't matter whether it's an artery or a vein. There are three distinct regions, layers, walls, tunics, whichever you want to call them. Starting from the outside and working our way in, there is the tunica externa. Some older textbooks will call this the tunica adventitia. This is the outermost layer, and it's composed principally of connective tissue, areolar connective tissue. And so it's going to have a lot of elastic fibers. It's going to have a lot of collagen fibers. And its principal role here on the outside of each of these examples here is to serve a structural function and a protective function. Moving inward, the next layer is the tunica media, and this is composed of smooth muscle cells, particularly in arteries and their smaller counterparts. The tunica media is really important because it is the actions of these smooth muscle cells that promote the dilation or the constriction of blood vessels. And you can see here the middle layer in each of these examples going from a large artery down to a very small, what's called arteriole, that they all have this tunica media and that the thickness of that tunica media varies with the size of the blood vessel. The innermost layer of any blood vessel, regardless of whether it is a artery, a vein, even if it's a capillary, is called the tunica interna. Some books call it the tunica intima. Means the same thing. You'll remember from our study of the heart that this is continuous with the innermost lining of the heart called the endocardium. We've also used another name for the tunica interna and the tunica intima, and that's the endothelium. I'm sorry that there are all of these different names. It's just the way life is with anatomy. Very frequently there'll be multiple names for the same structure, and you just got to know it because different people will use different terms. Now, there are a number of different types of blood vessels, right? We have the arteries, we have the veins, we have the capillaries, and I want to cover each of these three general types one by one. Let's tackle arteries first. Okay. Arteries, again, carry blood away from the heart, and they vary in size from extremely large arteries, such as your aorta and pulmonary trunk, to much smaller, what are called arterioles, which I'll talk about in a minute. So starting up here with the largest type of artery, these are what we call the conducting or elastic arteries. Both of those names should give you a hint as to what they do. They're involved in conducting large amounts of blood. Again, the examples I gave you were the pulmonary trunk, the aorta. Other examples are things like the common carotid, the subclavian artery, common iliac artery. Those are all examples of these conducting arteries. They conduct blood to major portions of the body. They're also called the elastic arteries because they have a lot of elastin fibers present in them, and this promotes a great deal of ability to recoil when stretched. This turns out to be a really important characteristic. Think about the aorta, which is directly connected to the left ventricle of the artery. The aorta has to withstand some incredible changes in pressure. Every time the heart beats, in systole, it's squeezing blood out of the left ventricle and into the aorta at a relatively high pressure. When that ventricle then goes into diastole and relaxes, it's then the aorta is experiencing a much reduced pressure. And so this elastic quality allows the aorta to expand when the pressure is high and then to recoil when the pressure drops. And what this does really is it helps to mitigate or reduce the impact of the sudden rise in pressure that's associated with systole. And then when the pressure drops during diastole, the recoil of that large artery helps to slow down how fast the pressure drop occurs. And what this does over time is it helps to even out the blood flow so that instead of getting these high changes, high spurt, high pressure followed by a low pressure drop, by the time we get out to the arterioles, the blood is flowing at more or less a constant pressure, and that's because of this expansion recoil. Expansion recoil is helping to even that out. The next size down in terms of the arteries are what are called the distributing or muscular arteries, both of which give you a hint as to what they do. They distribute blood to specific organs, and they tend to have a goodly amount of smooth muscle tissue, so they're often called the muscular organs. These are very vasodynamic. In fact, of all of the types of arteries, they are the most vasodynamic, meaning they have the greatest ability to dilate and constrict. And again, that's because they have the greatest amount of smooth muscle tissue. If we look at the superficial distributing arteries, the ones that are, in other words, located pretty close to the skin, these are the ones that serve as pressure points for controlling severe bleeding. So things like the femoral artery is, you can use that as a pressure point to stop bleeding in the legs. The next size down are the arterioles. These are the smallest of the true arteries, and they're a major site of regulation and distribution of blood. In other words, the arterioles are where we decide where the blood goes. Do I deliver it to this capillary bed or do I not deliver it to this capillary bed? You'll be interested to know that at any one point in time in your body, approximately 75% of your capillary beds are closed off and not receiving blood. It's the job of the arterioles to determine when a particular capillary bed should be delivered oxygenated blood and when it should not. If we didn't have these arterioles determining when a capillary bed should receive blood, in other words, if let's say all the capillary beds were receiving blood at the same time, you would not have a sufficient volume of blood in your body to perfuse or to deliver blood to all of those capillary beds. So we have to keep most of the capillary beds closed at any one point in time. There's not enough blood to go to all of them at the same time. We're going to learn about something called a meta-arteriole, which is a related term, in a little bit when I get to talking about capillary beds. For now, though, let's talk about capillaries in general. Capillaries are unique among blood vessels in that they only consist of a single layer. Remember, I told you typically there's those three layers, the tunica externa, the tunica media, the tunica intima. Well, in capillaries, there's only a single layer, and that's the endothelium or the tunica intima, meaning that these capillaries are a single cell layer thick. They are just made of simple squamous epithelial tissue, and here is a capillary in cross section. You can see that the diameter of this capillary is about the same as the diameter of a red blood cell or an erythrocyte. This means that erythrocytes oftentimes have to squeeze and fold themselves in order to make it through these capillary beds. We will learn later that that's an advantageous aspect because it assists us in having good gas exchange of carbon dioxide with oxygen. The major job of the capillaries is to deliver oxygen and pick up carbon dioxide from the tissues and to deliver nutrients, things like amino acids, glucose molecules, other nutrients such as vitamins and minerals. So we have, again, as I said, only a single cell layer thick in these capillaries, and of course always associated underneath any epithelium is that basement membrane layer. Now, what is a capillary bed? Well, a capillary bed consists of a arteriole, a network of capillaries, and an outgoing venule, or a really small vein that collects up the blood after it passes through a capillary bed. You might be interested to know that we have about 1 billion capillaries in the body, and that a cell, most cells in the body are no more than 0.2 to 0.4 micrometers in distance away from the closest capillary bed. So most cells in the body are very close to a capillary bed. There are exceptions. Remember, things like cartilage tissue is avascular, tendons and ligaments are avascular, the cornea of your eye is avascular, your epithelium is avascular. So those are exceptions. But generally speaking, most cells in the body are associated very near to a capillary bed. And there are in a capillary bed somewhere on the order of 10 to 100 capillaries. Now remember, I told you that most of the time capillary beds are closed. The anatomical feature responsible for keeping a capillary bed closed is something called a precapillary sphincter. And in this picture here, there are precapillary sphincters associated all along this midline blood vessel. This midline blood vessel is called a thoroughfare channel. And most of the time, blood just goes through the thoroughfare channel straight from the arteriole into the venule, because usually the precapillary sphincters along that thoroughfare channel are closed. Now, the region of the connection point between the arteriole here and the thoroughfare channel here is a little segment of vessel right here called the metarteriole. Metarteriole. There we go. And the job of the metarteriole is to simply connect an arteriole with the thoroughfare channel. There are different types of capillaries. depending on their role in the body. By far and away the most common type are what are called continuous capillaries. This is what we see in most places in the body. And continuous capillaries are just made of endothelial cells connected together. They often have narrow little intercellular clefts. That means little spaces between adjacent cells. And those intercellular clefts allow for the movement of solutes out of the blood and into the surrounding tissues. We will learn more about this process in a couple lectures from now when we get to capillary exchange. A less common type of capillary is the fenestrated capillary. Fenestra means window. So fenestrated capillaries literally have little windows or little holes that go right between two cells. Right between a cell. Okay, so in an individual cell, you can have little pores or windows, little holes in that cell. This is not between cells, this is within a endothelial cell. And those little holes or windows facilitate the more rapid passage of solutes. And fenestrated capillaries are usually seen in places where we have to move things very rapidly into and out of the bloodstream. So fenestrated capillaries are what you find in the hypothalamus, in the anterior pituitary. They're found in the kidneys, in the small intestines, etc. A specialized type of fenestrated capillary is called a sinusoid. And really all a sinusoid is a fenestrated capillary that is irregularly shaped. Rather than having a nice circular shape like most fenestrated capillaries would have, sinusoids can oftentimes have a flattened out shape such as this sinusoid here from the liver. These guys they have an irregular shape to them. They tend to have a lot of fenestrations or holes. They tend to conform to the shape of the organ space that they're associated with, and we see them in places like the bone marrow and the liver. So we've talked about the different types of arteries. We've talked briefly about the different types of capillaries. Now we need to finish up our discussion by talking about the different types of veins. Veins vary in size just like arteries vary in size. There are very large veins and there are very small veins. We still have all three layers present, the tunica externa, the tunica media, the tunica intima. One unique feature of veins that makes them very different from arteries is the size of the lumen. The lumen in veins, relative to their outside size, their outside diameter, is much larger. So veins on average have larger lumens than similarly sized arteries. And there's a reason for that I'll get to when I get talking about this question down here. The ability to make an incision or a puncture into a blood vessel, such as when you're taking blood, drawing blood from a vein, is called phlebotomy. So a person who draws blood is called a phlebotomist. Let's start from the capillary bed and move our way upward towards the heart, and we'll look at these veins as they increase in size. The smallest type vein, the one connected directly to a capillary bed, is called a venule. Again, it still has all three layers of tissue. They're just very thin. Veins in general have much less smooth muscle tissue than do arteries, and so they are not as vasodynamic. In other words, since they don't have very much smooth muscle tissue, they do not do a very good job of dilating and constricting. It's really not their deal. Remember, the main job of veins is to carry blood back to the heart. The next largest size vein above the venule are the medium-sized veins, and they collect blood from a number of different venules, and then many medium-sized veins will join together to form large veins such as the superior and inferior vena cava and others that are similarly sized, and they return the blood back to the heart. The blood pressure in veins is much lower than the blood pressure is in arteries. One of the reasons for that is, think of the pressure gradient. The point of highest pressure of blood is right when it exits out the left ventricle and into the aorta. As the blood travels through the systemic circuit, pressure drops. It just does, because as passing through these larger and then smaller and smaller and smaller sized arteries, there is friction. Okay, blood cells are bumping up against the wall of the arteries. That's considered friction, or what we call resistance, and that's going to diminish pressure. Okay, so by the time we get to the capillary beds, the pressure is very, very low. It has to be low in those capillaries because the capillaries are only a single cell layer thick, and if the pressure of the blood were very high, it would just shred, rip apart those capillaries. So on the return route back to the heart, by the time we're exiting out of a capillary bed and into a venule, the blood pressure is extremely low. And if the blood vessel were relatively small in diameter, it would make it very difficult for that blood to move. So this is why we have larger than typical lumens in the veins. The larger the lumen it is, the easier it is for blood to move through it. And since the blood's already at low pressure, by increasing the luminal size, we can facilitate easier movement of that blood on its return journey to the heart. By the time I get to the right aorta here, the right atrium here, blood is at its lowest pressure of anywhere in the body. Okay, if you think about this, this makes for a pressure gradient, doesn't it? We have high pressure in the aorta, much lower pressure in the superior and inferior vena cava, and as a result, we have a pressure gradient, and this assists in the movement of the blood through that systemic circuit. A similar logic applies to the pulmonary circuit. Couple more points about veins and venules to wrap this up. Veins and venules are uniquely different from arteries for a number of reasons we've already said. They have a larger lumen, they have very little smooth muscle tissue, and as a result of that and the fact that the blood pressure in the veins is very low, the challenge of veins is to get blood returned to the heart, particularly in instances where we're going against the flow of gravity. So, for example, let's say you're a vein in the lower leg. It's very difficult to oppose the pull of gravity and move blood upward towards the heart. As a result, we have some structures in place to assist in the movement of the blood back to the heart, and a key one among these is the internal valve. Veins and venules have within them at intermediate segments all along the length of the vein and venule, they have these little things called venous valves. And those venous valves allow for one-way flow of blood, just as valves do, so that, for example, if we're looking in a leg here and looking at a vein in cross-section, we can see that when blood passes through a valve, if gravity pulls on that blood back downward, you can see here that this valve closes off after the blood moves through it, and as a result, the blood cannot drop back down to where it had been. This process is assisted by skeletal muscles on either side of the vein. So every time these skeletal muscles contract, they squeeze blood up through the next venous valve. And then when the muscle relaxes, the blood drops back, but it can only drop back to this level because the valve prevents it from dropping further back. This explains why, for example, people who stand in one spot for a really long period of time can end up fainting. That's because the movement of not contracting and relaxing your leg muscles means that you're not assisting the return of blood to the heart, which means you're not getting blood back to the heart and not to the head. When you have inadequate blood flow to the brain responds by inducing dizziness and syncope, this fainting process that gets your body back on a level so that it's horizontal with the ground, and thus it's easier to return blood back to the heart and to the head. When people get varicose veins, this is a direct result of having these valves get stretched outward and become incompetent. So what happens then is, imagine if I have an incompetent venous valve, well, blood is going to begin pooling in the region where that incompetent valve is. And this over time can become visible, particularly with veins that are located very close under the skin. This is what we call varicose veins. Hemorrhoids, by the way, are one type of varicose veins. It's just varicose veins located in the region surrounding the anus. Now, there are some other mechanisms by which we get blood back to the heart through the veins. Not only do we rely on those venous valves, but we do rely on some other helpful strategies. Remember, we talked about the fact that there's a pressure gradient between the aorta and the superior and inferior vena cava, so that because pressure is highest in the aorta and pressure is lowest in the superior and inferior vena cava, there is the pressure gradient facilitating the movement of blood from the aorta and through the veins. There's also this skeletal muscle pump I just talked about, where the contraction of skeletal muscle tissue itself helps to squeeze blood through the veins, and in conjunction with these venous valves prevents it from falling back downward again. Your diaphragm in and of itself, the contraction and relaxation of your diaphragm below your lungs, also helps to produce a pumping motion that helps move venous blood upward. Gravity can assist in the movement of blood back to the heart if we're talking about places like your head and your neck and your shoulders. So we can use gravity to help us. And then there's this thing called cardiac suction. In cardiac suction, what's really happening here is after the ventricles contract, the collagen and elastin fibrous skeleton of the heart has the ability to recoil, to kind of pull the ventricles back open during diastole. That creates almost a suction kind of effect called cardiac suction. And that kind of suctioning effect of the ventricles also helps to pull blood from the atria into the ventricles and assist in the movement of the blood back to the heart. Enough on that. Next up, we're going to tackle the concept of resistance and blood pressure.